I would like to greet all our distinguished guests here, all our retired members of the NBPGR family, the ex-directors of NBPGR, and the whole NBPGR family to this 45th Foundation Day of ICAR NBPGR. I greet you all on this joyous occasion and acknowledge the gracious presence of the speaker for the 8th Dr. Harbhajan Singh Memorial Lecture, Professor Paul Gebs from Department of Plant Sciences, College of Agriculture and Environmental Sciences, University of California, Davis, and our chief guest, Dr. T.R. Sharma, Deputy Director General, Crop Sciences ICAR, our guest of honor, Dr. D.K. Yadav, ADG Seeds, ICAR, and Dr. Kuldeep Singh, Director NBPGR. And I would request Dr. Kuldeep Singh, sir, to kindly extend virtual hospitality and introduce the speaker of the 8th Dr. Harbhajan Singh Memorial Lecture.
Thank you. Thank you very much, Ratmanji. Good morning to all of you. Namaskar सबको और मेरी तरफ से सबको 45वें फाउंडेशन डे के बहुत बहुत बधाई. So I congratulate you all on the 45th Foundation Day of National Bureau of Plant Genetic Resources. Our today's chief guest, Dr. T.R. Sharma, Deputy Director General Crop Sciences. Our today very special invited speaker, Dr. Paul Gebs. Our former directors of NBPGR, Dr. Rai Rana, Dr. P.L. Gautam, Dr. B.S. Dillon, Dr. S.K. Sharma. Our other two former directors may not be able to join us because of their other commitments, Dr. Paroda and Dr. Bansal, but they have sent their good wishes. AD Saab, Dr. Yadav Saab, all heads of the divisions, all our peers who have served NBPGR in different capacities over several decades, those who laid the foundation of NBPGR, many of them are here with us today. I welcome you all my colleagues from NBPGR, its regional stations, all because we have sent this invitation to several people across the country, several universities, all ICAR institutions. So I welcome directors of all the ICAR institutions. I welcome all the participants from the state agricultural universities. In fact, we have very limitation of time, and I have the pleasure of doing two duties this morning. One is to briefly apprise you all, especially our peers, the progress made for the last 1 year at NBPGR, and we'll be making very brief of it. And then I will have the pleasure of introducing today's speaker. So we all know that 2020-21 was a COVID year, but despite that, the institution kept on working. And if I go by the record, 25 explorations were undertaken despite the COVID, and as many as 1,400 new extractions have been added to our germplasm, and more than 30% of that are crop sizes, 35% exactly in this year. We have identified at least 2 new taxa during this 1 year's time. Gap analysis is an important thing, and we have the instructions that we must complete the gap analysis of all the crops. And this year we have completed the georeferencing of 16 crops and have the georeferencing of them right now with us. Exchange was very, very important. We all know that due to COVID, it has been impacted severely across the world. But despite that, we could clear 125,000 samples of germplasm and international nurseries around the world. So these are two divisions doing exchange as well as the quarantine. We took special time ensuring that the seed reaches our clients at an appropriate time. There's no delay in that time. That was what my colleagues at NBPGR did, worked overtime to ensure that seed reached them in time. We had a responsibility of facilitating the export of germplasm as well. And this year, we have exported almost 13,000 samples. So that means the quarantine of that was very important before it reached the other side of the world. We did that one as well during this time. During this year, 2020-21, almost 26,000 accessions were characterized under field conditions. Those are primarily for the descriptors, for dust correctors, and evaluation of the important traits. As a result, several new genetic stocks have been identified, both for biotic stresses, abiotic stresses, as well as the nutritional traits. And a good number of them have already been identified with different markers to make them better usable. If we see this year's progress in germplasm, adding more germplasm to our gene bank, almost 7,000 new accessions were added to gene bank this year, making it a total of 452,000 accessions in the long-term storage of the conservation. So this is seed storage. Likewise, for cryo, 134 accessions were added. For in vitro, 31 new accessions were added, thus making our cryo gene bank to around 12,000 and in vitro gene bank around more than 1,900 accessions which are being conserved. To add to that, more than 3,000 DNA samples have been added to the genomics DNA bank in this year as well. We have a responsibility of DNA fingerprinting for the varieties which are to be released by the national system. And this year, we have been able to fingerprint 125 new varieties of 25 agri-horticultural crops in the country. 23 consignments of the GMOs of papaya and soybean arrived in the nation. We did analyze those as well. We have been given the responsibility to really develop the protocols for identifying the presence of the GM in food products. And we have been successful in developing the protocol for about 23 different products, including oil, where we can now analyze the presence of the GM in those. Several of my colleagues, they have been conferred different awards during this year. Dr. Gurundarjeet Randhawa was awarded with Punjab Rao Deshmukh Outstanding Women Scientist Award. Dr. Rakesh Singh, Dr. S.C. Dubey, they have been conferred with different awards. So this year, we have been able to publish 97 publications in peer-reviewed journals, 4 books, 2 books edited. 25 popular articles, and so on. So we made a lot of progress during this year, but we were unfortunate as well to have lost a couple of our colleagues during this year because of the COVID. We express our sincere thanks to all of them for their contributions to NBP. This is in brief the progress made during last 1 year. Now I will have the pleasure to introduce today's speaker, Our today's speaker is Dr. Paul Getz. Dr. Paul is a distinguished professor at the Department of Plant Sciences in the University of California, Davis. Paul did his PhD from the University of Wisconsin in plant breeding and genetics in 1984. Paul has an interest in several areas. The more important ones for us, which I would like to share with you, is his main thrust is in teaching and outreach programs in the study of crop agrobiodiversity for the purpose of conservation and application of breeding programs. His primary area of focus is on the physiology of beans. 5 of them, he is working on those. And he is a global leader when it comes to the beans, the genetic resources, the evolution, and the conservation of it. Paul is currently leading several global projects. The few ones which I will really mention are the California Dry Bean Advisory Board, Lima bean and garbanzo bean breeding. He is leading genomic recombination landscape of common beans in relation to drought and heat tolerance and other traits of agronomic importance. He is leading another Crickhouse Trust-led project, African Bean Consortium. Their research activities at the UC Davis are in support of the ABC breeding program for multiple disease resistance. He is leading another program on evolution and changing environment, genetic architecture of adaptation outside centers of domestication of Physiolus vulgaris. He's a very, very liked teacher by the people. And this I'm telling based on my experience with several students, that he's one of the best teachers in the university in the Department of Plant Sciences. And the important courses we teach is evolution of crop plants and plant genetics. All has 237 peer-reviewed publications, and he has participated in more than 300 international seminars and symposia. He has so far guided 16 master's students and 25 PhD students. Paul is on the board of several important leading journals of the world, and a few to quote is that he's associate editor of Frontiers in Plant Sciences. He's associate editor in Genetic Resources and Crop Evolution, and he's scientific coordinator of African Bean Consortium. Paul has been rewarded by several awards, and a few to mention are the Chancellor's Award for International Engagement at UC Davis in 2018, Agropolis Lewis Massachusetts Prize for Discipline Scientist 2017, International Scientific Prize for Agriculture and Food 2017, Frank Mayer Medal for Plant Genetic Resources by the Crop Science Society of America in 2015, Calvin Sterling Biodiversity Memorial Lecture Award by the Crop Science Society of America in 2014, D.Y. Morrison Award of USDA ARS American Society for Horticultural Sciences in 2013. Paul is a fellow of the Crop Science Society of America and American Society of the Ground. I know there's a limitation of the time. I cannot go beyond that one. So these brief about the progress made by NBPR during the year 2021 and a brief introduction of Paul. I invite Paul for delivering the 8th Dr. Harbhajan Singh Memorial Lecture. The floor is yours.
Thank you very much, Kuldeep. It's a real pleasure. I thank you very much for this very generous introduction, and it's a real pleasure and a big honor for me to be here today with you. I think the institution NBPGR is really well known around the world for its excellent work in germplasm conservation. And so for me, it is an honor to be with you. Frankly, I would have preferred to interact personally, but given the circumstances, the Zoom will do. uh, but certainly to visit some of your stations and so on would have been very much of interest to me. I should now actually share my screen, which I will do. Can you see my, wait a minute here. Can you see my screen here? The first slide, it should say challenges to the utilization.
Yes, the slides are visible. Please go ahead, Paul.
Oh, very good. So let me go then right into the talk here. Now, if all the buttons would work, that would be great too.
Just click again on the slide, please. And then you can use your arrow buttons or some buttons for moving ahead. Just click on that. Yeah.
Here. So I thought that I would just kind of highlight a few of the experiences I've had with genetic resources conservation and utilization. I really started, I got off to a good start because I became a research associate at a genetic resources unit of CIAT, which is one of the CG centers, just like you have ICAR in India. And I worked on Phaseolus germplasm. That's where I really started. Both wild and domesticated, and in interspecific crosses between Phaseolus species. From 1981 to 1984, I did a PhD research at the University of Wisconsin, as Kuldeep mentioned, and I studied the variation for seed proteins. And out of that work came the findings of a double domestication in common bean, Phaseolus vulgaris. And that was probably, according to Jack Harlan, the first time that somebody had unequivocally demonstrated the double domestication on molecular level. After 1985, I moved to California, first at Riverside and now at Davis. And in 2012, I added the responsibility of the grain legume breeding program, mostly in Phaseolus, but there is also a small amount of work on chickpea. So that has made me familiar with both gene banks as well as the use of the genetic resources conserved in these gene banks. And it seems to me that the major challenge of any organization, but especially of gene banks, is how can we improve the use of the genetic resources collections in these gene banks. It's often said that the gene banks are not used very much, for which I disagree. And I'll mention here at the left of the screen an example of one of your compatriots, Harry Upadhyaya of ICRISAT, where he calculated the number of accessions, for example, of groundnut, both domesticated and wild, 142, led to the development of more than 8,000 breeding lines. So you might say, well, 142 is not very much, but I would argue that that is precisely the value of a gene bank, is to provide that genetic diversity, and through the selection work of breeders, you develop a value-added project. And so I'm not that concerned with the actual numbers, but rather the availability and making these genes available to the breeding community and thereafter to the public. The same thing is for that study on the right here, a Chinese study, and you look at here, the right here, the percentage of the germplasm involved in the breeding of the improved lines. These numbers appear to be fairly low, but again, they are there and are available. If the numbers were be much higher, we wouldn't need a breeding program actually, and that is unlikely, given what we know about the characteristics of land races, of wild relatives, and so on. So the issue is not, to me, are these genes, are the gene banks really used? They are, and they are very useful. To me, the issue is more how can we increase their efficiency? Now, from a breeding standpoint, I like to show this pyramid here coming from a paper by Jim Kelly, who is a fellow bean geneticist and breeder at Michigan State University. And he and two of his students published a paper on the breeding for yield in common bean. And this pyramid is actually quite illustrative because the breeding activities really involve elite by elite crosses normally, and that's what breeders prefer. That's where they can make the fastest progress from selection. But occasionally there are new strains, new traits that are necessary. And so I say that figuratively breeders have to descend the pyramid and go to the bottom, and that's where the gene banks are. And conduct then intergene pool over the specific crosses or access crop wild relative. And then going back up the pyramid then to introgress these traits from the gene bank into the elite by elite crosses. And so that is really the challenge. How do we increase the efficiency of this whole genetic conservation in gene banks, the pre-breeding, and then eventually the breeding of improved cultivars. And a lot of what you will hear today is really about pre-breeding rather than breeding elite by elite, actually. So I thought that I would focus on the 6 major challenges I see in gene bank utilization. The first one is what I call the black box. What's in the gene bank, really? Kuldeep provided an impressive list of new traits, new accessions that had been identified. But how do we get there? How do we make this work more efficient, this discovery work more efficient? The second one is what I call lost in the woods. We have received germplasm from gene banks, and I'll provide more detail about that. And very often the germplasm, understandably, is not adapted, but to the point where we cannot really evaluate this germplasm. So it stays there, and I will propose kind of a strategy to really address this issue. The third one is the sampling issue, and especially the representativity of a collection. This is a moving target. Collections evolve, especially by addition of new germplasm, but how do we know that it is really representative? And then the fourth one is I contend that we need to be more specific how to target our conservation and breeding operations. Which populations, which traits? This is, for example, not just drought tolerance, but what traits specifically are involved in drought tolerance? What type of drought stress are we talking about? And so which environment also plays a role. And then the fifth one is the availability of all these data in suitable databases. And lastly, but not leastly, is the situation with international treaties and intellectual property rights. How does it affect the interchange of germplasm and the use of germplasm? That is an important issue, and it's unresolved for that. And I'll mention an example to which we have contributed that dealt with erroneous IP rights on a germplasm imported in the US. So let's now talk about the experimental model. You will have heard that I work mostly on Phaseolus. It is, from the standpoint of the study of genetic diversity, an extremely interesting genus because a lot of the genetic diversity has been shaped by domestication. And the genus itself, out of the 80 species here, and I'm citing here 3 references here that describe these 80 species, 5 have been domesticated. And 2 of them have been domesticated twice, actually. And so you see here the different, the 7 domestications with their respective wild progenitors. And we recently published a chapter in a book on population genomics edited by Dr. Omra Jora of New Brunswick University, and it's authored by Travis Parker. You will hear his name cited more often. He is a postdoc now in my lab, discussing the various aspects of these 7 domestications. Now, the domestication here is kind of interesting, primarily because we have different distributions. You see here the distribution of common bean, and I will come back to that, from northern Mexico to northwest Argentina. This is a distance of about 10,000 kilometers. You have lima bean, the wild progenitor here, the yellow dots, also a similar distribution. Runner bean is somewhat smaller, and then the most limited distribution is for the wild year bean, here a few locations in Guatemala. Okay, and so you have these different degrees of domestication, which allows us to answer questions about the effect of domestication on the genetic diversity. The species also have very different adaptations. You have, for example, tepary bean, which is of increasing interest because it comes from northwest Mexico, southwest US, and is very adapted to heat and drought stress, for example. In contrast, for example, runner bean, which is a high altitude, cool, and wet environment, for example. So there is all sorts of adaptations, all sorts of reproductive systems, from allogamy to autogamy, and so on, actually. So the first question I would like to address here is that kind of black box. What's in the collection, really? And most of the research in Phaseolus has focused on common bean because that is economically the most important species, but also it is also the most domesticated species. It's grown for its dry bean, but also for its green bean. And that variation actually appeared from this distribution, which is again shown here from northern Mexico to northwest Argentina. Now, several markers have been used to unravel or understand the genetic relationships among all these accessions. And it turns out that all of these Phaseolus species originated in what is called now Mesoamerica. You could call it Mexico and Central America. That is really the center of origin of the genus. All of the domesticated Phaseolus species have at least one domestication in this Mesoamerican area. So we see here, this group here all have a distribution in Mesoamerica. But there is this particular group here that has a distribution in the southern Andes, and this small group here in green that has a distribution in Ecuador and northern Peru. And so this research has come to the conclusion that actually there were rare long-distance dispersal events from the Mesoamerican area into the Andes. And this is dispersal is bird-mediated. And the green area here is a dispersal event that took place about 400,000 years. And the purple area here is a dispersal event that took place about 100,000 years. Now, the green area here is of interest because it includes sequences that are ancestral, but they don't exist anymore in the area of origin of the genus and of the species here in Mexico. So we are kind of in this strange situation of having ancestral sequence outside the ancestral region. And that is certainly a motive to conserve, but also the age on the order of hundreds of thousands of years, means that we have genetic diversity in the Andes that is qualitatively different than what's found in Mexico. And I'll give you an example later on of disease resistances that are different here in the Andean area compared to the Mesoamerican area. So I illustrate this example. I use this example to illustrate how important it is to understand the genetic relationships in a species, and especially of its wild progenitor, to understand the overall genetic diversity, but also the domestication patterns, okay? Now, if you look here to the right of the slide, this is principally about the domestications. There were two domestications in common bean, one of them here in central Mexico, and the other somewhere in the southern Andes. So the orange area and the purple area gave rise to domestications. The other areas, the blue area and the green area here, were not involved in domestication directly. And you see this here reflected in this graph here. You have a split down the middle. To the right, it is the Mesoamerican area. To the left, it is the southern Andean domestication. And each of them has its wild relatives that are more closely related to the respective domesticates. And that, again, is a confirmation of what we have seen before since 1986, that there are two domestications. Now, what is the consequence for that? For genetic conservation, it's clear that we need to conserve both areas. Even though the Andean area is derived from the Mesoamerican area, as I said, qualitatively, it is different. We also have, generally speaking, two different breeding programs, one to address the Mesoamerican cultivars and the other to address the Andean cultivars, okay? So here what you see, it's the same type of study, but involving primarily lines that are included in breeding programs, for example, at CIAT or in some of the African countries. And you see, again, that split down the middle, which indicates a double domestication. Here it's to the left is Mesoamerican, to the right it's Andean, but you see also further subdivision. For example, you see land races. You have here a group of Andean land races. This group here is a group of Mesoamerican land races from southern Mexico, and they fulfill a very important role as donors of disease resistance. Okay. You see also breeding objectives represented. This group here represent the activities of breeders in the Midwest of the United States who have converted varieties with a prostrate growth habit into varieties with an upright growth habit amenable to direct combining, for example. And then you see also certain ecogeographic races that are represented here. You see this group here are the ecogeographic race Mesoamerica. It is typically a low altitude, hot and humid. Whereas here we have this group here is an Andean ecogeographic race, the race Nueva Granada, which has often a high frequency of upright cultivar, fairly large seeds, fairly elongated. And I'll give you an example later on. You can also look at what is the diversity that is present in farmers' fields. We did a study in a region of Uganda, of Western Uganda, Hoima County. It's a fairly small county, but we analyzed 190 farms. Were able to secure a sample of their bean diversity. And you see here the diversity that we analyzed with SNP markers in the context of a broader representation of the germplasm. And the red branches here are the diversity that was present among these 190 cultivars. And I would say that that is quite impressive. This is diversity maintained at the farm level. If you analyze then these individual lines, we see that if you look at all the samples, they contain both Andean and Mesoamerican types. And among those farmers that were involved in a participatory varietal selection exercise by CIAT Africa, you see the effect of these participatory activities in the increased diversity figured by the red dots here. And so these participatory activities increase the diversity at the farm level. What is already quite diverse. Another sign of the importance of farmers in the selection of diversity is this so-called seed ngufu, which is a selection made by several farmers for a very particular type of variety and a red mottled seed type. And they appear to be able to maintain the purity of this particular farmer variety. Now, I wanted to transition. Most of these, what I've talked about the black box, were genotypic evaluations, but clearly we have to combine this to the phenotype. The genotypic evaluation provide us with an idea of the genetic relationships, but not necessarily of the agronomic traits or the phenotypic traits. So here, this is an example of a transition to the phenotype, and it illustrates the pattern of coevolution with pathogen. I see that somebody is raising, Mukul Kumar has raised his hand. Should I stop here or answer the question? Well, until I get an answer, I will continue. So what you see at the left side of the screen is an evaluation. What was collected were wild beans from Mexico, Ecuador, and Argentina. So these were Andean. This was the intermediate group, the green group I showed you, and then Argentina, which is an Andean, so Mesoamerican intermediate Andean. And you collect both the seeds of these wild beans as well as pathogen strains from the same plants in, again, Mexico, intermediate, and Andean area. And you evaluate whether there is a compatible reaction, which means actually a susceptible reaction on the part of beans. And the black squares here show the compatible reaction. And it shows that actually these Mexican wild beans are primarily susceptible to the strains of the pathogen from the same area. And so this is true for Mexico, true for the intermediate area, and true for Argentina. So there is a coevolution between the pathogen. Now, what is the practical application for that? It is precisely that if you want to have a resistant Argentinian material, an Andean material, where do you go for resistance? Well, you go here. You go to the Mexico, which has no susceptibility to the same pathogen. Okay, and vice versa. If you want to have a resistant Mexican genotype, you need to go to Argentina. Okay, so you need to be able to make crosses between the two gene pools, which is definitely possible. Why do we have this reaction? The same collaboration with French workers at the University of Paris showed that there are clusters of resistant gene. There's a large one on chromosome 4 of beans, and that cluster has generated both Andean and Mesoamerican resistances. Okay, and so you see major genes, CoY, CoZ, and Co9, but also quantitative traits indicated by the boxes here at the level of leaves, seeds, and pods. Okay, and this cluster actually predates the separation 19 million years ago between soybean and Phaseolus. And so again, it helps us understand to study where do we go for genetic diversity. Clearly, this example here at the left shows that we need to collect the entire distribution of wild beans and not just the one in Mexico that has the highest molecular diversity. We need to also look at the diversity in the Andes. Okay. Let me then also continue. I mentioned mostly genotypic evaluations, phenotypic evaluation in terms of disease resistances. I'll give you here 3 examples where we are trying to increase the efficiency of phenotypic evaluations. The first two examples are for growth in general or photosynthesis traits. And using drones, you can measure the growth rate of the canopy at the various stages. Especially, we did it for the early vigor, early growth vigor. We think that grain legumes suffer from generally poor growth and low speed of growth in the initial phases. And so Travis Parker used drones to actually follow the growth. He also conducted a genetic study. He developed a recombinant inbred population between a slow grower and a fast grower and was able to identify then QTLs. And clearly this approach can be followed in germplasm, even an increase to follow the growth, the speed of the growth of the different germplasm accessions. I'm involved right now in an experiment, mostly with physiologists in my department, who look at drought tolerance in a magic population. Troy Magny is one of the colleagues here, and he erected this tower here with cameras at different heights, and they can then follow, measure photosynthesis related traits of these different plots. So we have here a static as well as a dynamic or non-static measurement of plant traits at a high throughput. The third example here is conducted by Kimberly Gibson, who is one of my PhD students, and Emily Bick, who is a former graduate, an entomologist, former graduate of UC Davis. She works at the University of Copenhagen now, and they are working with a startup company who has developed these sensors here that can follow insects in the field. And we are trying to identify differences or resistances against the what is called Lygus hesperus. It is the major pest of lima bean in California. And you can actually train these sensors to identify the Lygus insects, but also the predators of Lygus or the hyperparasites. So there is both a genetic component and a cultural component in terms of biological control, actually. And I believe that these approaches are absolutely essential if we are to increase our efficiency of phenotypic evaluation. Both the genotypic and phenotypic evaluations ought to be applied to germplasm collections so that people can actually get away from this black box phenomenon. So I would then turn to the second issue that I'm raising, and this kind of the lack of basic adaptation in germplasm collection. This is a perfectly normal situation. You would expect this, but how do we get ourselves out of this problem? So I define what I would call basic adaptation. It's, can the plant complete its cycle and for seed propagated species at least produce some seeds, for example, go through the vegetative and reproductive parts of the cycle. We are not actually dealing with what I would call advanced adaptation. It is the optimization of performance. That is a breeding process that comes afterwards. But it would be great if we could already get this, just the basic adaptation. And I'll give you an example. We received several years ago a collection of lima bean from the CIAT Genetic Resources Unit. Only 10% of these 300 accessions flowered and set at least some seeds. That means that 270 accessions did not flower. This is a tremendous loss, and I would say it's almost a tragedy that we can't evaluate these 270 accessions, for example, for these potential Lygus resistance. So what is needed, actually? I think it's a different approach. It's not novel. It's what I would call germplasm conversion. People have done this before, like in sorghum, for example. And what you need to do is to map and preferably identify these basic adaptation genes. And they are very often domestication genes. So you have flowering and photoperiod genes. You have growth habit genes. Another aspect here of this lima bean is that they're all climbing types. Well, that's good in certain types of agriculture, but in something like California, we would like to see bush types. So how do we convert a climbing type into a bush type? We also would like to avoid seed dispersal for wild types, and so lack of pod dehiscence. And in California, they like, for whatever reason, they like white seeds. So we need to have a gene for white, or an allele for white seeds, for example. So as I said, it has been done before, but the approach here is not to take adapted germplasm and introgress a trait of interest like insect resistance. It's actually take the unadapted germplasm and introgress a limited set of adaptation genes, like photoperiod, for example, like bush growth habit. These would be 2 genes only. And then once you have selected for those, for the rest, you can continue doing pedigree selection. And so this would make the germplasm much more available. So we conducted this study on domestication syndrome some time. This was published in 1996. And from a scientific standpoint, it is very heartening to me that now the genes that we had mapped but not really isolated, that they are coming out but in the location that we had predicted based on an RFLP mapping, a crude RFLP mapping. And so it is really, to me, very satisfying. And this actually shows a summary of these genes that have been isolated and really in the location that we had predicted. We mapped here isolated the first gene for determinacy. Others have, with our materials actually, have mapped and isolated a gene for photoperiod sensitivity. Recently, they identified a gene for seed dormancy in exactly the location we had identified as a QTL. You have the gene here for white seeds. It's the P locus mapped on the chromosome 7. And then we are now working on elucidating the genetics of pod shattering. And so again, this is, so some of these genes are the genes that we need to integrate into the germplasm to make it amenable to evaluation. Okay. I will skip in the interest of time, but as I said, it's you take a domesticated, unadapted germplasm and you cross it to a domesticated, adapted germplasm that has these individual genes for adaptation. It should be a fairly simple process. It can be done with markers because we have the sequences, we have mapped them. And so we'll see how that turns out. The third issue I want to talk about is the sampling or representativity of the collection. I'll give you the example of wild common bean again. The collections are collections are a dynamic concept, constantly evolving, mostly by addition. So how do we know that, and since when do we know that wild common has such an extensive distribution over 10,000 kilometers? Well, it started in the 1940s in Argentina and Guatemala. Why such a disjunct distribution? People should have scratched their head. In the 1950s, you see some individuals timidly collecting some materials. Then in addition to Guatemala, in southern Mexico, also in Colombia, in Peru, as well as Argentina. In the 1960s, there was a major effort in Mexico, and you see then a large number of wild bean collections made in Mexico. In the 1970s, the pace starts picking up in different countries, and it really breaks open in the 1980s with a large number of collections. You see then that this distribution is taking shape. And there are gaps in this, but they are likely to be real gaps in the distribution and not gaps in the collection. Okay? Then the 1990s, you see a continuation of that. And then after the year 2000, it stops. And it stops. We have now overall distribution, but there is almost no collection being done. Why is that? We can discuss this, but the fact of the matter is that, yes, we know the distribution of the wild common bean, but should we continue collecting, and what should we be collecting? I have some ideas about that. So this is a table that reflects that, and you see the 2 decades here, 1980s, 1990s, with a tremendous number of collections. The type of data that were used were initially roadmaps, which, believe it or not, sometimes can be state secrets or military secrets in the Andes. We used molecular diversity to argue that we needed to fill some of these gaps. We have the global weather data to indicate what type of climate do these populations live in, and now more recently species distribution models also. So we can also use then, in terms of studying the representativity, this is in a sense the genealogy of common bean. You see here a lineage that started out probably more than about 1 million years ago. You have a first dispersal event towards Ecuador and northern Peru about 500,000 years ago. A second long-distance dispersal event about 100,000 years ago. And then that lineage ends up forming the Mesoamerican gene pool. This dispersal event is the Andean gene pool, both of which were domesticated and eventually disseminated around the world. So that is the view, what we know about the diversity. We can then evaluate, for example, what is the USDA core collection, which supposed to represent a representative subsample. How is it representative? Well, what I've done is to take the same graph and I've grayed out the populations that are not represented here. And you see that there are significant omissions in this core collection. Now, this is not a criticism of this particular core collection. It was one of the first one that was set up at the beginning of the 1990s, but it's definitely should be updated with what we now know about the genetic diversity and the genetic materials that are represented in the USDA Phaseolus collection. Okay. Now, I mentioned that I had some ideas about what should we collect nowadays. A lot of the collections so far have investigated the distribution of a taxon like wild common bean by following roads in the potential areas of distribution. This is the case here. You see a road here, and lo and behold, we found wild common bean here against this rock face. We decided then to look around, and lo and behold, yes, there was another popular series of plants here. Interestingly, these are wild common bean growing on wild maize, just like the cropping system that we see very often around the world now, with beans growing physically on top of a maize, domesticated maize. You also see here another example of wild beans growing on wild maize. We see that the population actually is not just along the roads, which is a favorable environment for these wild populations, but further down away from the road. And this, we really have very little idea of what diversity grows away from the roads, actually. You see also here another wild plant. And what you see here is in that field, this is maize, but it's really a hybrid between domesticated and wild maize, probably backcrossed towards maize. And this is then a form of wild squash, all in this fairly small field. So there is tremendous diversity. I've also mentioned the example of collecting pathogens from wild populations. And so the model that I propose is to move from a geographic exploration to an agroecological exploration, so that because crops are part of agroecosystems. The crops don't grow on their own. That is an invention of the modern industrial agriculture. Crops are, like here, growing with other crops like maize and squash. So you have crop populations, you have crop interactions. They in turn have pathogens and pests, but they're also beneficial microorganisms, both endogenous and exogenous microbiomes. In the case of beans, the rhizobia are very important. And so I would argue that if and when we continue explorations, we should take these other organisms into account, and the facilities of gene banks ought to follow this evolution. Let me then go to the fourth area, is the targeting of traits. I think that which population, which trait, which environment, there is an inherent difficulty in evaluating wild species, wild relative. I'll take the example of wild beans here. There is a yield evaluation. It's very difficult to evaluate yield, seed yield, for example, in a wild relative. They are not selected for that, unlike domesticated plants. There are different selection regimes altogether. Wild beans disperse their seed explosively at maturity. How do you want to evaluate yield in that situation? There is also a stronger population structure and a more localized adaptation than in the domesticated descendant. And so it is very difficult to choose, for example, high-yielding wild populations. That is almost an oxymoron. It's nonsense. So we set up an experiment with Jorge Berni, a former student, that involved actually, we wanted to see if we could identify drought-tolerant wild beans. So we took 3 populations of wild beans along a rainfall gradient from Durango in the north, 500 millimeters, to Huehuetenango in Guatemala, almost 2,000 millimeters. The populations were crossed, 1 backcross and 4 selfings, and there was a selection for earliness and photoperiodin sensitivity. And then once we had these populations, they were tested under well-watered and terminal drought. This is what the population looked like. You see here in the field, the green area, it's caused by lateness in spite of the selection, and that is a disadvantage compared to the domesticated breeding part of the nursery, where you see materials already well advanced. Okay. So the populations were genotyped, were phenotyped, and the central result is that we were able to increase the yield of the domesticated tester only in the crosses that involve the 2 populations from the driest area. The population from the wettest location did not either increase or decrease the yield. Now this to us was a positive result, because it says, yes, we can increase yield under drought stress conditions, and we can actually choose specific populations to that effect, in spite of the difficulties in evaluating directly these wild populations. Okay, so the choice of population is important. We now know how to do this. We can use GIS, for example, to select the driest environment. The testing environment is important, and the choice of traits. As it turns out, earlier experiment had shown that wild populations in dry environments have a deeper root system compared to others that come from wetter areas. Also, these wild beans, when you subject them to drought, they continue growing in spite of the drought, unlike the domesticated types. So we have two specific traits here, the root system as well as the continuation of growth, that are specific traits that can be used to screen for both wild and domesticated germplasm. And so it's really the specific trait. It's not just drought tolerance. No, it is specific drought stress tolerant traits. I also believe that in the future, we will have to take care of consumer preferences more than we have done. This is the question here is, you see common bean is the most consumed bean species, but you have tepary bean and lima bean. Each of them have some resistance to drought. The question is, will the consumer be agreeable to the consumption of these other bean species? Okay, with that we don't know. But we work already within common bean to develop improved heirloom-like varieties that have increased yield and virus resistance. These are completely different from the normal commercial types and expands actually the use of the diversity contained in germplasm collections, whether they are public or commercial collections. So I'm getting now towards the end of my talk. There is a problem, an issue that I would like to raise, and that is the databases. I don't think that there is really a satisfactory database that really combines all the data and can be interchanged. I've heard with interest that Kuldeep mentioned about the DNA fingerprinting. What I would like to see is the databases that where you can look at or can switch around from phenotypic to genotypic data back and forth. An example of where the benefit when we have these phenotypic data is, for example, the gene bank at CIAT has the photos of a large number of its accessions. Now, to breeders, this is extremely useful because the preference of the consumer, and so the breeding target, an important part, are the seed types, the color, the shape, the patterning, and so on. And we have used this information actually to test for the representation of the core collection, as I discussed before. We have also used pod shattering data from the USDA to find that the Durango race in northern Mexico is really tolerant to pod shattering, which makes sense given the aridity of the environment. So we need more integration of different types of data. And I would argue that the research community of a crop, for example, the Phaseolus community, ought to have this partial responsibility of curation of the data as well. And then finally, the constraint here is these intellectual property rights and international treaties, and to what extent do they affect the free interchange of germplasm? Maybe we won't get back to a free interchange, but at least a fairly easy interchange so that research towards applying the genetic diversity can proceed. I will give you an example where I was involved with that. At one time in 1999, a patent and a plant variety protection certificate were awarded to an individual who claimed to have developed a yellow bean variety. And it's this particular, he gave him the name of Enola, but you see that in Mexico, they had similar varieties, and as well as in Peru. And actually the Mexican bean breeders had developed a new commercial class resulting from the cross between a azufrado, so a sulfur bean, and a canario, a Peruvian canario, And so that generated that new class, Azufrado peruano. The bean community was really, I would say, befuddled, if not upset, about this type of patent. I took it upon myself to develop a small research project, and we fingerprinted these small collection of beans containing both yellow and non-yellow beans. And you see here the marker of choice at that time, and that's 20 years ago, were AFLPs. Some of you may remember those, but it allowed us to fingerprint these materials. And the result of that was, again, you see here, principal coordinate analysis, again, the split Mesoamerican versus Andean. Because the cross involved these Peruvian Andean Canarios, you see most of the yellow bean were actually Andean. And Enola here has the same fingerprint as AP87. So we were able to identify a potential source of this variety, which was not an original breeding effort. Through a course of multiple appeals, eventually the court that is just below the Supreme Court of the United States decided to cancel many of the claims made in that patent. And in effect, the patent became null and void, actually. And so I felt that we had contributed to this decision, which was really almost a fraudulent claim to original breeding product, actually. But and so I was very pleased to hear Kuldeep mentioning that a lot of the fingerprinting going on in the collection, because I think that that is an important point to maintain the ownership or sovereignty over this this germplasm. So in conclusion then, and I thank you for your attention and for your patience, I mentioned the black box problem. We need faster and better genotyping and phenotyping of the collection. The problem of lack of basic adaptation, I propose this germplasm conversion, where you see marker-assisted introgression of a limited set of domestication or adaptation genes. The representativity, we need to continue evaluating the representation of these collections, and there ought to be a new model for exploration based on agroecological sampling. The targeting issue is we need to make an informed choice of accessions and populations, specific phenotypes or traits, and also the environments of origin and testing. And that way we'll be able to make a better choice of the germplasm and selection in the progenies using that germplasm. The database problem, I think that that is a continuing problem. Some databases are better than others, but I really, we need to improve on that. And I think that the respective crop communities can contribute that through their curation. The thicket of IP rights and international treaties, I think it prevents and slows down the active interchange of germplasm and associated information. And I think that it's too bad. I think that there is a sense of urgency about feeding an increasing population throughout the world. And so that's what I had to tell you. And I'm certainly open for questions. Thank you very much.
Thank you so much, sir. Thank you for taking us through the evolutionary relationships to phenotypic evaluation, to genotyping, to mass, to domestication, sampling problems and databases, the whole gamut of research in PGR that you have provided us. I think we are all truly enlightened by your talk. I think sir has already mentioned that he is ready to take a few questions. we should be a little brief as we are pressed for time.
May I? Sunil Archak from NBPGR.
Yes. Dr. Sunil Archak.
Thank you. Thank you, Professor Paul, for this fantastic lecture. It was just like two scale mass with rhythm, flow, and variety. Thank you very much. You have thrown a dice of 6 faces, and we know that how to identify priority. My specific question is on the centricity of the crop diversity. Centricity, the question of centricity. People are coming up with various theories, particularly in barley. Particularly in barley, they have shown that it is not mono or oligocentric. It can be a continuous geographic region. So you have shown some glimpses of a similar kind of non-canonical centricity in phaseolus. So what is your take on for gene banks to work on some kind of identifying these centricity issues so that we actually focus on proper collections as well as resolving these disputes between different countries? Thank you.
I'm not sure that I fully understood your question. I'm sorry about that.
May I repeat? Yeah, may I repeat if you would like?
Yes, please.
Yeah, thank you. This is regarding the centricity of crop origin and diversity.
Yeah.
Yeah, you have shown some glimpses of that in phaseolus, that it need not be canonically monocentric, as we have studied in textbooks. It can be multiple places. People have shown in barley that a very typical genomic exercise in barley has shown that it can be a geographically continuous region.
Yeah.
So what is your take on this centricity research by gene banks? How can we work on that?
Well, you know, I think what you should strive for is really to be able to combine the germplasm in the case of barley, it's both the one of the Fertile Crescent and the one the further east of maybe what is it, Tibet or the northern India. They ought to be both accessible to the user, just like in beans, both the Mesoamerican and the Andean ought to be accessible. Now, I know that the treaty, the CBD, for example, says that sovereignty lies with the individual countries, and that may seem to, that's the problem I'm alluding to, is that it may seem to prevent or slow down the interchange of germplasm. That is what we need to fight, actually, I think. Because if you were to conduct a breeding program just for Andean bean or Mesoamerican bean, or in the case of barley, you would be missing out on a significant portion of the germplasm. And so I know that it's becoming every time more complicated. It's not just the seeds. Now they are also talking about the information, for example, traditional knowledge, but also the DNA sequence information becomes an issue. I think that all of these things make things more difficult, and they have not promoted the use of germplasm. And that there is a cost for that, actually. It assumes that the reasoning is that if you can control it, you're going to benefit from it. I question that assumption.
Yeah.
Because if it stays in the gene bank, it's not going to be put to use. I know that maybe some people will disagree with me, but I think the best germplasm is the one that gets used and evaluated.
Thank you. Thank you, Paul.
Thank you so much, sir.
I'm not sure if I answered your question. I'm sorry, but maybe we can have a discussion later on.
If there are, Dr. Kuldeep Singh, sir. So please unmute yourself. I think sir is not able to
unmute yourself.
Okay,
Yes.
now it's good. That's good. Thank you. I think Archik, you need to ask the organizers, the host, that he really needs to allow us to unmute. Okay, thank you, Paul. Thank you very much for taking us for a beautiful journey from gene banks to the breeding lines, how we can really do it. You have shown a beautiful method, beautiful way how things have been done for beans, and especially in one of the continents now. How do you think that if we really have to do it at a global level, how can this be replicated for different crops at a global level? Because ultimately we need to see the overall diversity, especially the ones which you have shown that the resistance genes and some important genes may be found across the domestication sites or so. So, for example, if we take a crop, major crops like wheat or rice, or for that purpose, where the germplasm is very, very large, how can we really do it in such crops globally?
Which crops are you referring to?
If I talk of major crops like rice and wheat or maize.
Yeah. It's the first time I hear that phaseolus would be an example for rice and maize. I'm joking. I think we have a pretty good idea because we started off studying the wild progenitor, that we have a good understanding of domestication. And as I said, in beans, common bean, that was the first example where we fairly conclusively could demonstrate a double domestication, multiple domestication. I think in rice it's more complicated. I'm not sure why. I say you have one domestication, 1.5, 2, or even 3 domestication, depending on the paper you read, actually. But understanding that will make you understand how the genetic diversity is organized. At the same time, you have to collect these other organisms, like these pathogens, to understand the diversity of the pathogen and then the interaction. We did not do that only in the Americas. We have done this also in Africa. The so-called ABC project, the African Bean Consortium, the students study the diversity of the pathogen as well as the bean host. And so they know what the diversity is of the pathogen, especially of the virulence. And they know if there is resistance among the local germplasm, or do they have to import germplasm from the gene bank. So I think that the model is perfectly extensible to other crops. It starts with trying to understand the genetic diversity, actually. And both at the wild level and at the domesticated level. The paper of 1991 that describes the ecogeographic races in common bean is a citation, has become a citation classic. And I don't mean to brag about it, but it's how useful that concept has been for bean breeders or anybody who is using bean diversity. And it's based partly on molecular markers, partly on the distribution of phenotypes, like disease resistances, growth habits, adaptation, and so on, actually. So from 1991, so that was fairly early on, actually. So I think that it's extensible to other crops.
Thank you so much, sir. Dr. Shashni, if he's able to unmute, please.
Yeah, thank you. Thank you, Dr. Paul. It's a wonderful lecture. I have a simple question to ask. When we predict the timeline of domestication, so I know there are different prediction programs, and we go for the evolutionary significance, evolutionary aspect of the different species or the DNA. But how far do you think these are correct when you predict? Because you have not seen how the crops are being domesticated and they are being grown in different regions. Thank you.
Yeah. Well, on one hand, I would say there is always an element of uncertainty because we weren't there. But I think that there is now increasingly a combination of DNA sequence and also archaeobotanical remains that can be dated. And through accelerated mass spectrometry, you can actually, you only need a fairly small amount of biological material to date the remain, actually. And there are some striking cases where people have been able to see a correspondence between the DNA data and the carbon-14 dating, actually. So I think that it's possible within, being certain level of precision, actually, which is probably on the order of several hundreds of years, even 1,000 or 2,000 years. Actually. Now, maybe you want something more specific, but I think personally, I'm satisfied with that. And we see it fairly consistently across crop in a certain region. I would be more concerned about the absence of archaeological or archaeobotanical remains, because the absence of evidence is not the evidence of absence. And many times, the absence of archaeobotanical remains doesn't mean anything, actually. So hopefully that will maybe provide some elements of answer.
Thank you so much, sir.
Manjusha, I think Professor T.R. Sharma perhaps would like to intervene.
So, Sir Dr. T.R. Sharma, please.
Good morning to all. Good morning, Dr. Paul. It was really indeed an excellent presentation which you have made.
Thank you.
And while referring to co-evolution of host and pathogen, have you looked at the pathogen population of common beans and what was that population? How did you look at that?
You mean of the pathogens?
Yes.
Yeah. So we did it both molecularly as well as phenotypically by cross-inoculating the beans on which they were found, from which they were isolated. And so there is a one-to-one relationship. That is very important because people, that's how we found that you have Mesoamerican strains on Mesoamerican beans and vice versa for the Andean. Most people skipped the isolation, the one-to-one relationship actually, and just dealt with it geographically. The molecular marker we chose, for example, right at the beginning, were like rapid markers. We wouldn't be using these nowadays, but there are other markers from sequences that are fairly conserved, actually. And so it's both genotypic as well as phenotypic. And likewise for the host. It's markers to see, is it Andean or Mesoamerican? but also the susceptibility to the pathogen.
Okay.
And
Thank you, sir.
if I may, another element is that we did this not in the Andean and Mesoamerican centers of diversity. We did it outside, in Brazil and in Africa, because I reasoned that in those outside, you have both gene pools, host gene pools represented. In Brazil, you have 20% Andean, 80% Mesoamerican beings. In Malawi, it's the reverse. You have 80% Andean, 20% Mesoamerican. So the pathogen, in a sense, can choose if it's going to infect a Mesoamerican being or an Andean being. Okay. And so leaving that opportunity. So there you have the possibility, and we see that there was a preference for a certain gene pool and not just the two equally. So that was an important distinction. I'm sorry for interrupting, ma'am.
No, sir. I thank you again. And I think there is one question by Kostav Panda on the chat box. I think sir will read it and reply to it. And since we do not have too much time to interact now, I think I would ask all the participants to kindly chat and directly talk to Dr. Paul Getz. We are now moving on to having a release of our publications by
Manjusha, one second.
yeah.
Professor Paul has been standing ever since he has started talking. We are all sitting. We are all sitting in our rooms comfortably. Paul is standing there. Professor Paul, the studio, we request you to stay with us till these couple of things are released, and one of which is the point which you raised, one of the database kind of thing. Thank you.
Yeah, I don't mind standing. That's how I teach. I find that I'm more dynamic than if I'm sitting.
So we would like you, Dr. T.R. Sharma, sir, Dr. D.K. Yadav, sir, to please release a few publications. If we were here, I mean, having a in-person meeting, I think we would be able to meet the developers and the authors of these publications. I would request all the authors to kindly, show their videos. At least we can have a look at your faces while we are doing this. First in the lineup is the folders from NBPGR Regional Station Jodhpur. These are in Hindi. One is Til ki Jaiv Vividta ka Aay Srijan mein Upyog by Vijay Singh Meena, Rashmi Yadav, Neelam Shekhawat, Kartar Singh and Kuldeep Singh. The other one is Ek Nazar Mein, Rashtriya Padap Anuvanshik Sansadhan Bureau, Jodhpur. This is by Kartar Singh, Vijay Singh Meena, Neelam Shekhawat, Veena Gupta and Kuldeep Singh. The third one is Padap Anuvanshik Sansadhano Ka Prabandhan. This is by Neelam Shekhawat, Vijay Singh Meena, Kartar Singh, Veena Gupta and Kuldeep Singh. The next one is from Regional Station Bhuwali. It is an information brochure on cultivation of kiwi in Uttarakhand hills. This is again in Hindi, and it has, the authors are K.M. Rai, Mamta Arya, Anuj Sharma, P.S. Mehta and Veena Gupta. Another one is genus Sesamum L in India, an illustrated guide for species identification. This is a booklet that is based on the information of exploratory trips, morphological studies undertaken in the experimental fields, and herbarium studies in reputed herbaria. And the authors are Dr. K. Pradeep, Suma Parimalan, Rashmi Yadav, Swati, and Reshma. And the next one is PGR Clim. It is an application which is going to be used for climate smart PGR management. The developer for this is Dr. Sunil Archat, officer in charge, AKMU, NBPGR, and we would like all the dignitaries to kindly launch this application, and I would request Dr. Archak to kindly speak about it.
Thank you very much. Thank you very much indeed. This is PGR CLIM. This is called as PGR CLIM. It is an online tool to achieve climate-ready gene bank. This is the first initiative from our side. Because to be climate-ready, gene banks have to follow a twin-track approach of designating pre-adapted germplasm for immediate direct use or varietal development, as well as to identify vulnerable areas for collection and conservation of germplasm. We had a CCAPS funded project, PG CCAPS funded project from 2011 to 15, where we georeferenced more than 60,000 indigenous successions in 10 target crops. What we did was to link the climate data and the passport data, and carried out clustering, climate matching, identifying vulnerable areas, designating pre-adapted material, and eventually we developed the climate maps. And a lot of work has been done on that. But that generated only static climate maps, which were in fact inaugurated by DG in 2016. But then we thought that these must be available to researchers to carry out their own analysis in an interactive fashion, which is why in ICAR National Fellow Project, we developed climate maps in ArcMap 10.1. But that was also, again, one has to host it on a GIS server. And here comes the role of Indian Agricultural Statistical Research Institute, ISRI. The purpose of making PGR CLIM available to researchers, that was possible because of ISRI. The team showed a proactive approach in redesigning the application, the latest version of ArcGIS, and then they have hosted it on 10.8 version of the ArcGIS server on Krishi Geo portal. I'll just quickly thank people who were involved in this. The study was possible because of the proactive initiative of Professor Kailash Panchal, at that time the director of NBPGR, and Professor Pramod Agrawal, CCAP's regional director, as well as Dr. Prem Mathur, who was the regional director at that time in the Biodiversity. We all know that PGR work, plant genetic resources work, is a teamwork always, and contributions of all the past and present colleagues of NBPGR in collecting germplasm is acknowledged, without which this could not have been possible. PGR CLIM tool is an excellent example of collaboration between different institutes, national and international. At this point of time, it is between NBPGR, our team. I thank Anuj, Firoj, and Ratnesh, people there who are involved, as well as ISRI, Rajendra Prasad, Anshu, and Mukesh. Now, I request this to be shared, please, from NBPGR. I'll just quickly show in 2 minutes how does it look like. Vijay or Ratnesh?
I will stop sharing then.
Yeah, please. Would you like to share there, Vijay? Quickly. Thank you. This is how it looks like. We have multiple layers on here. We have both state level boundaries, national, district level boundaries, so that one can scan information from multiple layers and then take out information both as the administrative boundaries as well as agroecological regions. We have a soil map. We have predicted data of climate, both temperature as well as rainfall, past, present, and near future and far future. And we can select different crops. For example, here I would like to request people to, let's say, quickly work on. We have used 10 crops here, as well as different, let's say, in 2 of the crops, wild relatives we have used. This is a pilot project. We wanted to learn from this so that we can extend this work in future. So let's say we have selected here brinjal crop wild relatives. We select multiple layers, future, let's say and just run it, and we can see how we can take out the data. Once we can do that, all we have to do is to just select different layers and then export it to Excel. So the people who are interested to access the collection points, we use the geo codes from where these have been collected over a period of time as the surrogate points for their actual occurrence at this point of time. so that it helps us in finding out gaps, in finding out vulnerable areas today and tomorrow. Can we see in some other crops if they are ready? I would love to, in fact, demonstrate this in detail, but because of shortage of time, I'm going to share the URL on the chat box. I request colleagues to just test that one, then give the feedback. We have developed some analysis, but any analysis is limited by one's own imagination. So this tool is now available for everybody to run various kinds of analysis, identify gaps, then generate lots of data. Thank you very much.
Thank you, Dr. Sunil. We would like to have blessings of our past directors. In that stead, I would ask Dr. S.K. Sharma sir to please bless us. If not, sir is not able to unmute himself.
Just please let me know. We will see.
Yes, I think sir is trying to unmute.
I'll do that one. Professor Sharma, please unmute yourself, sir. Now you are able to unmute now. Yeah. Please.
Okay. Thank you very much. Thank you very much for giving me an opportunity to speak few words on the very auspicious occasion of the Bureau today. Dr. T.R. Sharma, the chief guest of the function, Dr. D.K. Yadav, the guest of honor, Professor Paul Gapps, Dr. P.L. Gautam, Dr. R.S. Rana, Dr. Kuldeep Singh, the distinguished participants, guests, scientists, and the staff of the Bureau. I take this opportunity to congratulate the director and the NBPGR team on this 45th Foundation Day of the Bureau. The lecture by Professor Paul Gapps was very inspiring. It was very educating, and indeed, this is an area which is of concerns to all the gene banks globally, and is also being asked by the funding organizations regarding the utilization of the extent of utilization of the genetic resources which are conserved in the gene bank. After leaving the Bureau during 2010, I was in touch with the Bureau. I remained in touch with the Bureau in different capacities, and happy to see the progress the Bureau has made in the area of the PGR management, be conservation, exploration, evaluation, genomic resource development, database development, and so on. And this has been reflected in the form of scientific quality of the annual reports, the other books, reports, documents, development of databases, and more important, the quality of peer-reviewed publications with very high impact journal, which I also mentioned in one of the meetings of the RAC. And more importantly, the awards and the honors which have been bestowed upon the scientists of the Bureau. And I congratulate you all, the awardees, on this occasion. The Bureau is recognized nationally and internationally in the area of PGR management. And I hope in years to come, the Bureau would be a leader, a torch bearer to other nations in the area of plant genetic resource management. And with a few words, I wish the Bureau, the staff, and the scientists a great success in years to come. Thank you very much.
Thank you so much, sir. I'd like to request Dr. Dhillon, although I don't see him.
Manjusha, Dr. Dhillon has a weak connectivity, so he has sent his congratulatory note. He won't be able to speak. So he has a poor connectivity at the moment. So you may.
Dr. Gautam, sir, please.
Let me see if he's.
Sir was there. We have to unmute him.
But, I can't see with what name
Dr.
he is.
Rana. Dr. Rana, sir.
Thank you. Well, I congratulate the Bureau as I see the progress report presented by Dr. Kuldeep. It is really commendable. I had been associated with the persons who conceived the conservation of germplasm in India and then established the Bureau as the lead center. What was really conceptualized was conservation under natural conditions with evolutionary process intact. Long-term storage or the gene bank was only thought to be as a duplicate safety. We always emphasize networking, regional stations, active germplasm sites, and we had started at that time about 60 such sites. Idea was that they should be grown in climatic conditions from where they were collected, and evolutionary processes are continuing there. We invite breeders to these sites and they pick up what they feel is useful to them. We also envisage crop advisory committees to have better coordination between the Bureau and the breeder. In that context, I would just suggest, possibly apart from the progress that has been presented, which I highly appreciate, we should also keep in mind that kind of network, which really was the basis of our effort that we anticipate. Thank you very much.
Thank you so much, sir. With that, I think we would ask Dr. D.K. Yadav, ADG Seeds, to kindly address us. We'd be honored, sir, to hear you.
Thank you, madam.
Thanks.
Good morning to all. I personally welcome for this 45th Foundation Day celebration of this National Bureau of Plant Genetic Resources. Honorable Dr. T.R. Sharma, Chief Guest for today's function, and our distinguished speaker of 8th, Dr. Harbhajan Singh Lecturer, Dr. Paul, the distinguished professor from UC Davis, former directors of the Bureau, Honorable Professor T.L. Gautam, Dr. R.S. Rana, Dr. B.S. Dillo, Dr. S.K. Sharma, the present director, Dr. Kuldeep Singh, the directors of the other institute, I can see Dr. A.K. Singh is there, Dr. O.P. Yadav from Kajri, Dr. Rajendra Prasad from ISRI, Dr. Shasni from NIPB, Dr. Suresh Pal from NIAP, Dr. Subhash Chandra and CIPM, and Dr. Arunachalam from Capri, and Dr. Uma from Banana Institute, and also our former colleagues from Bureau, Dr. Moria and Dr. Saxena, all the scientists from the various ICAR institutes and SAUs, and also Dr. J.C. Rana and Dr. Vasne from the CG centers, and all the staff of the National Bureau of Plant Genetic Resources. So first of all, my heartiest congratulations and my best wishes to the director and all of his staff of the Bureau on this very auspicious occasion. As we are all well aware that the rich genetic diversity is the key to the success of any genetic enhancement program, may it be in case of crops, animals, fish, and so on. ICAR has always given highest priority for the management of biodiversity, which includes the top varieties, livestock, fish breeds, and agriculturally useful insects and microbial species. And NBPGR has also played a very significant role in the country in the collection, conservation, characterization, and exchange of plant genetic resources to both public and private sector stakeholders. And the director has very elaborately presented about all these activities during his presentation. So as a result of these concerted efforts, the Bureau, our National Gene Bank at the Bureau today is the second largest gene bank globally, and we have now conserved more than 4.5 lakh germplasm accessions, and we are playing a very important role in the management and utilization of genetic resources, not only within country, but also we are providing the leadership internationally and have the collaborations with FAO and United States and other CG institutions. We held for past few years about the safety duplicates, and with the very specific interest of our Honorable DG Saab and DDG Saab, we have started to find out for establishment the National Safety Copy Plant Gene Bank. And for that already a national expert committee under the chairmanship of Honorable Dr. R.S. Paroda was made, and all our former directors, including Dr. Gautam and all senior peers, they were the part of that committee, and the committee has given one preliminary report, and based on that we have already initiated some process, and for that a specific provision will be made in terms of funding also in the times to come once we are finalizing all these modalities for establishment of this NSCPBG. And in this directions, our Honorable DDG has already written a letter to the coordinator of the Svalbard Global Seed Vault, Dr. Asmad Asdal, and we have sought some information, basic information regarding the this safety duplicate gene bank, and he was very kind enough to give some specifications. And also we have requested him to be on the expert panel to give the advice on the various issues of establishment of safety duplicate facilities. And I am very happy to share that he has already agreed for be a member on our expert panel for this safety duplicate gene bank. Further, to strengthen the various activities other than the normal routine institutional that funding, we created implemented a consortium research platform on agrobiodiversity during 12th five-year plan, and now it is in its third phase during the next plan 2021-26, which is already the EFC is in process. And this consortium that includes 60 institutes and state agriculture universities and covering a huge array of plant, animal, fish. And now we have included the microbes and insects also, which were deleted in the second phase. So that is also, that program is a very, I think, dynamic and a huge program which is going on. Earlier, the funding was a little bit less during last 4 years, 2017, 20 to 21, it was around 12 crore rupees, but now we have announced this funding for the next phase, that is up to 31 crores. And I hope that is one program which is, I think, giving a good visibility, particularly in terms of the characterization, evaluation, and regeneration of the various genetic resources. And during past phase, 26,000 germplasm accessions, they were characterized, evaluated, and regenerated. And now, in case of maize, around more than 11,000 accessions, they are already under process of evaluation at the regional IIMR's regional station at Begusarai. So that is one of the very important aspects. Being now we are the Asia's largest gene bank, and which was established at Rana has mentioned, that established during 1997, almost 20 years back. So it required a huge renovation, total renovation. And during 1819, we could provide funding of around 20 crores. And now this National Gene Bank is the state-of-art gene bank, competing with many of the best global facilities, particularly in terms of the gene bank. So that has already is in operation. The another aspect which has been given to Bureau, although this NRC DNA fingerprinting was already there, but the Council has given the responsibility for the DNA fingerprinting of all the genetic resources, including the released varieties. So they are also helping in that aspect through the PGR, that genomic research. And now we have submitted a proposal after having a long deliberation through the national system, NAS. And that proposal is already for the 30 field crops for their varietal identity, and that will be used for the genetic purity also during the various certification programs. So that is already with GAC, and we hope that in the times to come it will be coming. Digitalization is one of the very important aspect, I think, in the today's era. And already Dr. Archak has done a very good job, and I think most of the data that is now digitalized and that is online, and today's the new portal that has also been shown. But still, I think we need to have some more work on that directions. The barcoding of all the PGR in the gene bank that has been is being discussed for a long time, and I think some work has been initiated. So I think the barcoding in terms of the whole passport data and all the specific traits for which the germplasm is there, so that needs to be done on priority. And now where everybody is talking about the precision for all the aspects. So when we talk of the precision breeding, that I have been discussing with Dr. Kuldeep many times, that when we give a set of germplasm to any of the indenters, so we should have the that genomic sequences for the various markers which have to be used, so that the breeder, once he is taking that material along with the genomic information, he or she, they can go for validation, and then they can use that particular material for interrogation of the specific traits for that germplasm has been borrowed from or that taken from the NBPGR. So that type of, I think, culture will have to inculcate in the NBPGR with the help of the various commodity institutions, because the Bureau alone cannot do all those things. There is no doubt that the excellent achievements have been made, and we have a very successful past and historical achievements are there, but still on some of these aspects we need to work. The new lead which Dr. Kuldeep Singh's intervention has taken, that we have now a MOA with the National Medicinal Plant Board, and already the MOA has been signed along with the National Botanical Survey of India also, because that component was earlier lacking. And I think with this, that particular for the medicinal plants also, they will now be, the set will be conserved with the NBPGR. So with this background, and the today's celebration of Foundation Day by the ICAR NBPGR is very much in the line of the programs and the priorities of the government of India in general, and with the of the ICAR in particular, fulfilling all the global obligations following the various regulations internationally and nationally. I specifically have the I thank to the Professor Paul for his very, I think, comprehensive lecture, particularly on the challenges to the utilization of gene banks, and from starting from the very beginning to the present day technologies, he has elaborated very nicely and given the challenges and their solutions also. So thank you very much, sir, for accepting our request and being with us. My sincere thanks to our DDG Crop Science, Dr. T.R. Sharmaji, for that on this accepting our request for this gracious presence and agreeing to be the chief guest. And I am also very grateful to our honorable former directors and the other distinguished guests and scientists for attending this program. I once again compliment the director and the whole staff of the Bureau for organizing this program, having a very beautiful work which they have done, and look forward for more vibrant programs in the future also, and wish them all the best for their enhanced role in crop improvement programs across the country through their trait-specific evaluation program. Thank you very much, sir.
Thank you so much, sir. Thank you so much for guiding us. And I'd like to now request Dr. R.C. Agrawal, sir, DDG Education, to please bless us.
Thank you very much, Dr. Manjusha. Thanks to Honorable all previous directors, Honorable DDG Crop Science, and the present director, Dr. Kuldeep Singh. My good wishes to this great institute, which in a very short time has grown like anything. and has made its impact not only at the national level, but at the global level. Because I have been closely associated, so I have a special affection for this institute. And whatever we could achieve in life, that credit goes to NBPGR, because my all directors with whom I worked, especially I can see today Dr. Rana, Dr. Gautam, I can see Dr. Bansal, and many, Dr. Sharma, Dr. Dhillon, everybody was so supportive, everybody was so visionary that whatever we are today, it was just because of their visions, because of their hard work, which they just put. And it's really a pleasure that we are witnessing the progress today. We could see from the report of Dr. Kuldeep Singh, the lot of awards which have come to the scientists, and a lot of progress in a very, very short time. We could see from Dr. Sunil Archak's presentation. I want to just congratulate for launching such important software, the CLIM. So really, it's a matter of great satisfaction, matter of great pride for all of us. And I want to congratulate DDG Crop Science that his institute is growing like anything. And it makes us all proud. And we are really so satisfied with the progress of this institute. So once again, I compliment each one of you, and I congratulate each one of you on this important day. Thank you all. Thank you. Namaskar.
Thank you so much, sir.
Manju
I'd
sir,
like to read a
Gautam
message.
Sab has joined. No, Gautam Sab has joined if you want.
Dr. Dhillon has also joined. So if our chief guest would allow, I would request.
Yeah, please go ahead.
Sir, Dr. Dhillon, please.
No, Dr. Gautam is available quickly. I'll try to see if Dr. Dhillon is unmute or not.
Okay, Dr. Gautam, please then. So please bless us then.
Dr. Sharma, am I audible?
Yes, sir.
Yes.
Okay.
Yes.
Dr. D.R. Sharma, the DDG Crop Science and chief guest of today's function, the speaker of the day, who has given very interesting insight of germplasm enhancement, management, and characterization. My all previous directors of NBPGR, the President Director, Dr. Kuldeep Singh, and all our members of NBPGR family and other ICR institutions. The nodal point in ICR of the institute, Dr. Yadav. It is a matter of great pleasure for me to speak to you on the 45th Foundation Day of the Institute. I feel so happy that the tradition of celebrations of the Foundation Day, which we formally started in 1997, is being continued with great interest. And the very, the very fact that you are having the Harbhajan Singh Memorial Lecture by eminent speakers on this day added a lot of value to the celebrations. As Dr. Agrawal said, that this institute has done tremendous work, and we have grown, and much of our growth we attribute to NBPGR. Well, we have several memories and reminiscences. The time is short. I would not like to dwell on that, but it was my brief stint of about 3 years in NBPGR, which to me appears to be a very productive time. And all of you were so helpful in executing the programs of the NBPGR, because it was transforming from old gene bank building to the new gene bank building, and taking many initiatives, linking with institutes in India and abroad. It was a great task. I enjoyed it, and I really would like to congratulate all the people, the persons associated with NBPGR. There are many people who have retired, and they have contributed a lot. Everybody looks towards the NBPGR for good practices, and that is what is being maintained, and compliments to all for this purpose. I would not like to comment on the speeches which were made because some part of it I could not witness, but as the comments go, they deserve congratulations for excellent presentations. So best wishes to you all, and I join you in this very gala celebration of the 45th Foundation Day. My best wishes to you.
Thank you, sir. Thank you so much. We would just see whether...
Yeah, Dr. Dhillon is available, please.
Yes, sir, Dr. Dhillon, please.
No, I think there's some connectivity problem with that Dhillon. He has sent a message again. I think we can continue further.
I think sir has written congratulations to the NBPGR family on the occasion of 45th Foundation Day and best wishes for the future. Pray that NBPGR continues serving the nation and humanity. With that very apt message, I think I would like to request the Honorable Chief Guest, Dr. T.R. Sharma, DDG Crop Sciences ICAR, to kindly address this August gathering.
Thank you very much. Good morning to all. Distinguished Director of the Institute, Dr. Kuldeep Singh, my colleague from the Council, Dr. R.C. Agrawal, DDG Education, former directors of the Institute, those who have joined today, Honorable Dr. P.L. Gautam, Dr. B.S. Dhillon, Dr. S.K. Sharma, and Dr. R.S. Rana. Speaker of the day, Dr. Paul Gept, distinguished professor from UC Davis, my colleague from Council, Dr. D.K. Yadav, distinguished directors of different ICAR institutes, NBPGR family, ladies and gentlemen. First of all, I would like to congratulate you all on this very auspicious occasion, that is 45th Foundation Day of the Institute. It becomes more important when we have organized Harbhajan Singh Memorial Lecture by none other than Professor Paul Gept, who has given very important insight of genetic resources, how we should conserve, characterize, and utilize in plant breeding. I would like to mention here that NBPGR is a flagship institute of the Council, and we always look forward to see that this institute should grow greater heights and try to help them from the Council side. We all know that plant genetic resources, or NBPGR, has 5 different verticals. That is collection, conservation, characterization, exchange, and utilization of germplasm. These 5 verticals are very, very important and interlinked with each other. If one of the vertical is weak, then probably it is going to affect whole process of developing plant varieties. This particular lecture, which was delivered by Professor Paul, is also very important because we are celebrating the 75th anniversary of Indian independence to commemorate the monumental occasion by all the departments and ministry under the government of India. And we all are hosting various activities to celebrate Azadi Ka Amrit Mahotsav. In that context, ICAR is organizing a series of 75 lectures under the leadership of my colleague, Dr. R.C. Agrawal. And many of these lectures have been completed and delivered by very distinguished scientists across the globe. So this 45th Foundation Day of NBPGR and the lecture by Professor Paul is another important activity of this particular Azadi Ka Amrit Mahotsav. We all know that India is bestowed with the immense richness of agricultural biodiversity, including diversity in crop plants, wild plant diversity, livestock diversity, aquatic diversity, and microbial diversity. Among the 12 mega centers for the plant species diversity of the world, Mega Gene Center in India has great importance. Northeastern Himalayas and Western Ghats are the 2 out of the 31 global hotspots of the biodiversity that are present in India. The different gene centers in India possesses about 11.9% of world flora with 5,725 species representing 141 genera and over 47 families of higher plants. Let me mention here that Crop Science Division of Indian Council of Agriculture Research works on more than 85 crops or field crops and trying to improve different varieties of these field crops, for which role of plant genetic resources is very, very important. We all know that plant genetic resources are one of the essential components that holds the key to the very foundation of agriculture, as well as food and nutritional security for the world. During the last 4 decades, rapid progress in the field of agriculture was witnessed along with the collection, conservation, and sustainable utilization of plant genetic resources. The presentation made by Dr. Kuldeep Singh about the progress of the Institute during past 1 year is really commendable. And it's at this point in time, I would also like to mention that the utilization of plant genetic resources is very dear to my heart because I started utilizing plant genetic resources in interspecific hybridization from 1988 onwards, first in brassica and then in rice. And I think we have extensively used these wild species local land races, and different varieties of brassica and rice germplasm for identification of genes and their characterization, mining alleles from different land races, and orthologs from different wild species of rice. Therefore, I always see a great hope and always look for the conservation of these resources, genetic resources, which are a great reservoir of genes for different traits. As we know, the current status of National Seed Gene Bank is 4.5 lakhs accessions belonging to more than 2,000 species of agriculture as well as horticultural crops and their wild and weedy relatives. In addition, more than 1,900 accessions are conserved in vitro and more than 11,000 accessions have been cryopreserved. That's the great strength of NBPGR, on which all our biologists and breeders, they are dependent. If anybody is working on any biological problem in the country, I think their first source of material is NBPGR. Therefore, exchange of resources, genetic resources, is very, very important activity of the Institute, which is driving the biological research along with agriculture research in the country. We know that NBPGR need to strengthen the plant genetic resource network of the country, and there is a need to revisit and strengthen the national active germplasm sites with their greater active role in the management and utilization of plant genetic resources. Dr. Yadav has already mentioned that we have a CRP on agrobiodiversity with more than 60 centers across the country. And that CRP, Consortium Research Project on Agrobiodiversity, is becoming very, very important and significant and contributing a lot towards the conservation and utilization as well as characterization of genetic resources. However, in present context, international negotiations and the rapid advancement in the fields of molecular biology, biotechnology, and bioinformatics have led to the emergence of new legal, political, and technical regimes that dictate plant genetic resource management. The current barriers to free flow of plant genetic resources, increasing intellectual property.
Kindly unmute, kindly mute yourself, please.
Sir, Dr. Sharma, you are muted. Can you unmute, please?
Yeah.
Thank you.
Okay. The current barriers to free flow of PGR, increasing intellectual property right issues, and the vast potential of biological wealth yet to be tapped through bioprospecting and genetic engineering. And it has placed greater demand on nations to adjust to the global changing scenario. And our speaker, Professor Paul, has given an excellent example of how IPR issues can be tackled, and we can do fingerprinting of our germplasm. And I am happy to note that we have a very active DNA fingerprinting group working at NBPGR, which is characterizing with all varieties of different crops and germplasm using different types of DNA markers. We know that during the past couple of years, the Bureau has made great progress in the direction of plant genetic resource conservation, utilization, and characterization through a paradigm shift in its evaluation strategy of germplasm, which is being followed by National Gene Bank in various crops. And these crops like minor legumes, minor oilseeds, chickpeas, rice, and wheat are being undertaken through a funding from the Department of Biotechnology, Government of India. And I would like to congratulate Dr. Kuldeep Singh, the director of the institute, and his team of scientists for attracting more than 350 crore rupees through competitive grants to complete this great task of genotyping and phenotyping of germplasm resources of different crop species, which just now I mentioned. We know that the current emphasis of crop improvement programs is to develop cultivars having genes for resistance or tolerance to abiotic stresses such as cold or drought, and biotic stresses like diseases and insects. Besides that, quality of produce, water input use efficiency, and stability of the performance. There is also a growing requirement for a wider variety of agri-horticultural products with diversity in taste, color, and nutrition, etc., which are of high value in the market. There is a need to modify the descriptors for evaluations accordingly. I would like to stress upon here that we have to characterize our genetic resources, particularly major food security crops like wheat, rice, and maize for their nutritional status. If I take one example of rice, most of the micronutrients are only present in either in the seed coat or aileron layer. So can we find some genetic resources in which endosperm is enriched with micronutrients like iron and zinc as well as protein, so that the eatable products which we are getting from rice should be made available to the end user. This is one of the examples I am sharing here, but likewise in wheat, even in millets, and also in maize, most of these micronutrients are already are available in their seed coat or in the pericarp or aileron layer. So one has to identify the plant genetic resources across the crops are these quality traits which are becoming very, very important to overcome malnutrition in the country. You know, for the characterization and trait-specific evaluation of germplasm, we have at NBPGR wild garden facility for crop wild relatives at IRI farm. Central Instrumentation Laboratory has also been created. We have very good field gene bank and ISAPOR, and many other facilities are being created at NBPGR so that they can contribute towards the development of different species. I'm really happy to note that the scientists have published a very good number of peer-reviewed papers, but it should need to be now published in high impact journals so that we can show our strength, and that can only be possible if you develop networks across the institutions, crop-based institutions, and then try to use all available approaches for utilization of germplasm resources for the mapping, characterization, and cloning of genes, as well as dissecting of QTL to identify different genes present in the QTL. I'm also happy to note that various publications have been released along with a database on named as PGR Claim by Dr. Sunil, and this type of databases have to be enriched and should be made more users friendly for the use of the scientific community. Before I close, I would like to quote what Father of Nation Mahatma Gandhi has said in 1930. To quote him, respect earth and life in all its diversity. Recognize that all beings are independent, interdependent, and every form of life has value regardless of its worth to human. Unquote. On this occasion of 45th Foundation Day of NBPGR, I would like to say that this is the day of introspection, what we have done during the past year and what we can plan for the next year. And we have the blessings of all our previous directors and mentors. We can make use of their expertise in various activities and various programs of the Institute. And I am really blessed to know their suggestions and get their suggestions at this particular occasion so that we can diversify and make our programs more effective. I would like to congratulate again Director of the Institute, Dr. Kuldeep Singh, for effectively steering all the activities and programs of the Bureau. I think the utilization of germplasm, particularly in pre-breeding program, has been started by Dr. Kuldeep Singh because of his earlier experience at PAU. And I would like to mention here that pre-breeding activity is one of the most important programs across the institutes which we started last year. And for that, NBPGR has to contribute a lot. And for that, I am always looking for the help of NBPGR. I would like to extend my heartiest wishes and congratulations to all the officers and staff of NBPGR on this Foundation Day. And I wish that such type of lectures, which was organized today by Dr. Kuldeep Singh, should also follow in future, and we should try to learn from our international experts on such occasions where almost all institutes are connected. So thank you very much, Dr. Singh, for giving me opportunity to be here this morning and to be part of this very important program of the Institute. Namaskar, Jai Hind.
Thank you, sir. Thank you so much for inspiring us. Your work on mapping, cloning, and characterization of peak is now textbook material, and giving us points to ponder in our research in this short address. We are very hard pressed for time, and we know that we have all our retired colleagues with us with whom we would have loved to interact, but we are not able to. But in adversity, we also find some opportunities, so we can rejoice that all our regional stations, the 10 regional stations, colleague are all with us today, and we are having just one Foundation Day celebration rather than having 11 that we normally do. And with this, I'd like to request Dr. Kavita Gupta, OIC PME Cell, to propose a vote of thanks.
Thank you, Dr. Manjisha. Respected DDG Crop Sciences, Dr. T.R. Sharma, the chief guest of the program today, Professor Paul Getz, Distinguished Professor, UC Davis, former directors of NBPGR, senior retired faculty, senior colleagues, and dear friends joining us today for celebrating the 45th Foundation Day. I have been assigned the very pleasant task of proposing the vote of thanks. First of all, we wish to express our gratitude to the Honorable Dr. T.R. Sharma, the DDG Crop Sciences, for chairing today's function. Thank you, sir, for your kind support, inspiring words, and for your keen interest in utilization of PGR. We are extremely thankful to Dr. Paul Getz, Distinguished Professor, University of California, who is a renowned scientist, for his wonderful lecture on challenges to the utilization of gene banks. Sir, we are extremely fortunate to have got the opportunity to listen to you as well as to interact with you. We also thank Dr. R.C. Agarwal, DDG Education, for allowing us the use of ICR facilities, as well as for his kind remarks. We also thank Dr. D.K. Yadavji for his kind and gracious presence this morning, and also for his special remarks. I also want to thank our former directors, Dr. R.S. Rana, Dr. P.L. Gautam, Dr. B.S. Dhillon, and Dr. S.K. Sharma, senior retired faculty, for accepting our invitation and for their blessings. Special thanks to our director, Dr. Kuldeep Singh, who has been involved right from the beginning and who has steered the organization of this entire program. I sincerely thank my colleagues, Dr. Sunil Archak, Dr. Rakesh Bhardwaj, Dr. Sushil Pandey, Dr. Ravi Pamathi, Mr. Abhishek Srivastav, Mr. K.K. Sharma, Arun Sharma, Sadhana, members of the organizing committee, for meticulously planning and organizing this event. A special thanks to Dr. Manjusha for smoothly moderating the program, and to Shri Vijay Mandal for his logistics support. I also thank Mrs. Kanchan Khurana, especially for helping with the invitations. A special thanks to my colleagues in PME Cell, Shivangi, Vikas, and Sanjeev, for their all-out help and support. A big thank you to all the participants, senior colleagues from ICAR, vice chancellors of universities, directors of ICR institutes, participants from PPVFRA, CSIR institutes, MOEF, NBA, ICFRE, BSI, DRDO, and state biodiversity boards for their presence this morning. I also thank all the heads of divisions, OICs of units and regional stations, and the staff of NBPGR for their participation today. A very special thank you to all the students participating today. In the end, I wish to thank one and all who have contributed towards this webinar in one way or the other. Thank you. Over to you, Manjusha.
Manjusha, you are muted. Can you unmute?
Unmute yourself.
I think we are all done now. We thank you all the participants again, and we wish you a very happy Foundation Day, the 45th. I think it is our Ruby anniversary, as ma'am told before. So rejoice, everybody.
What?
Okay,
once again, special thanks to Paul for staying with us. It's almost 11:30 there, it seems. So thank you very much. So thank you very much to all of you. Thank you.